And we have seen an exponential increase, a geometric increase, in the number of base pairs we could sequence daily. Another fact of the genetic revolution has been the ability to pinpoint specific abnormalities in the human genome. This is a gene, CHF, which is involved in age-related macular degeneration. For those who get a little older, we tend to lose a little bit of focusing ability in the macula, and that's some of those patients had genetic abnormalities that predispose to that. In 2005, a single genetic abnormality was tied to that disease. Look what's happened in 2009. We have realized that this isn't a single disease, single point disease. It's actually a disease with 400 separate loci that inform that disease. So we are now beginning, at least in some diseases, to realize that it isn't a single point mutation in the human genome, but it's a complex of genetic abnormalities that contribute to the formation of that disease. Look at the cost of sequencing. When you try to sequence the human genome, it will cost you about $100,000 to do the whole thing. By 2010, that'll be down to $30,000 because of automation. And that process is going to be coming into how we look at tumor cells. We are now realizing that cancer cells are not a single population of cells, but a large population of diverse cells. Tumors are more than just cancer cells. In fact, they're small organs. They're systems. They're complicated systems that interact with their environment. Here's an example of that. The old thinking about tumor cells were that the cancer cell came from one cancer cell and that replicated itself. But we now realize that, in fact, when tumors grow, there is something about the interaction of those cancer cells with their environment. That interaction, how you get along with your neighbor, is what helps make that tumor cell unique. It grows in an environment. It has to grow in an environment. And prostate cancer goes from the prostate to the bone. Why does it bypass the liver, the lungs? There's something about the nature of prostate cancer cells that allows it to grow better in bone. Lung cancer, unfortunately, tends to go from lung to liver and brain, not to other areas. There's some genetic reason for that. There's some biological reason for that. It has to do with the fact that tumors grow in unique microenvironments. So we used to look at just the tumor itself growing, but we realize now that so-called benign tissues or benign cells like fibroblasts and endothelial cells that are where the vasculature comes in and perfuses the tumor, and other so-called benign cells inform the growth of the tumor microenvironment. When I was going to medical school, the fibroblast was the most malign cell in the laboratory. It was a cell that was involved in healing and not much else. So we didn't spend much time learning about it. And now we realize that the little old fibroblast is actually involved in multiple pathways. So cancer-activated fibroblasts are now critical to the growth of a cancer in its particular site. And we now have ways of looking at a number of different activation pathways for the fibroblast, and we'll be able to target those. So we're no longer just targeting the cancer cell itself, but the environment in which that cancer cell grows.